Good morning, everyone. On behalf of the organizing committee, I would like to thank you for joining this discussion on sourcing seeds for climate resilience during ecological restoration. We have a great program today, beginning with two speakers, Julie Ederson and Anna Bukharova, who have each done significant research on climate change and the effects of sourcing and commercial production on the genetic background of seeds. These will be followed by lightning talks and another featured speaker. Jennifer Ogle will highlight her experience putting together a network of local seed sourcing partners in Arkansas. Please feel free to ask the speakers questions in the chat window. Their presentations are recorded, and they are happy to answer questions as we listen. We have a large group registered today, so please keep your video turned off and your microphones muted to ensure that the network doesn't get bogged down. Use the chat function to ask questions, make comments, etc. Your questions and comments will be saved as part of the archive of this meeting. Everyone will be assigned to a breakout group so that we will have the opportunity for smaller discussions. Each group does have a moderator who will be leading the discussion and taking notes. Obviously, this is a complex topic, and all of the participants bring a unique experience and perspective to the table. We will synthesize today's conversations and make them available in the coming weeks. We hope that at least some of you will be willing to continue in follow-up groups. We will also be presenting a follow-up conversation at the virtual Wild Things Conference in February. Our featured presentations will begin shortly.
Good morning, everyone. Welcome to this exciting webinar conference that's called Growing Through Change, Sourcing Climate Resilient Seed for Ecological Restoration. My name is Julie Ederson, and I'm a professor at the University of Minnesota Duluth and also the director of the Institute on the Environment in Duluth. I am here today to tell you a little bit about the work that I've done over the years to study the impact of climate change on native species. And I'm just going to start out with this really cool model that was developed for Glacial National Park. And so you can see it's stepping over time. And in the first years, there is a description based on color of what used to be on the ground: rock, glaciers, grasslands, coniferous, deciduous, etc. Different kinds of vegetation based on old photographs and notes, and then eventually on satellite imagery, and then pushing into the future based on climate modeling. And so you can see really obviously with this little map what's happening by 2040 or 2030. We've completely lost the glaciers at Glacial National Park. I think here it's going to flip back to see what it was like in previous decades. And what you notice is that as it's warmed over time, the kind of vegetation that occurs in the park has gradually moved up elevation. And so species like this lupin or species like this pica are moving up and hopefully still having some place to persist on the mountaintops, whereas other species like this bear grass or an elk are displacing them across space. But there are similar patterns across the United States. So if you look at this USDA plant hardiness zone map, here is one from 1990, and it shows me up here in the frigid land of Duluth, Minnesota in zone 3, where you cannot get a tomato to ripen by the end of the summer. But over the course of time, things have really changed in Duluth. Now we're fully in zone 4. and I can grow tomatoes. But also, we're seeing gradual changes in the vegetation that's around us. And basically, we are having climate that's moving up latitude, and we're having changes in the vegetation that are also moving up latitude. But this really only happens if you have a continuous landscape, like in a preserved national park. But we all know that that is not the case across a lot of the land. And here's just an example from Stearns County, Minnesota, a depiction of pre-settlement vegetation based on bearing tree data. And you can see that all these yellow and golden yellowy patches are prairie. And now I'm going to switch to a contemporary map to show you how much of that is left. And in the new map, anything that's white is vegetation that has been completely degraded or destroyed by human behavior. So are you ready? Here we go. Boom! If you really like squint, you can find these little patches of prairie, but basically it is embedded in an agricultural landscape. And so the potential for populations to be able to hop, skip through dispersal across this landscape and move into a habitat that might be more appropriate is could be obstructed because of habitat degradation. So given this scenario, I actually have spent most of my career trying to figure out if plants can't move, do they have the genetic diversity to evolve or adapt rapidly enough to keep pace with climate change? And here are 3 species that I've worked on, and just really simply, I'll tell you that the answer for Camiocrista fasciculata is definitely no. They cannot evolve fast enough. The answer for Campanulostrum americanum is that some traits can evolve fast enough, like flowering time, but it causes maladaptation in many other ways that affect survival and fitness. I've also worked in this goldenrod species, which is a little odd because it has both diploid, 2 sets of chromosomes, and hexaploid, 6 sets of chromosome populations that are segregated across space. Hexaploids cannot evolve fast enough. Diploids can. But the bulk of evidence suggests that plants cannot evolve. They cannot adapt fast enough to keep pace with climate change. So what do we do? Well, in the latter part of my career recently, I've been thinking more about what we put in our restoration sites. You know, we're doing all this restoration, and we're just going to have to do more and more of it into the future. So what material should we use? Should we use local, or should we be thinking about using plant material that might be pre-adapted to the climates of the future, or at least catch up with how much climate change has already happened? So this is where I live on the shore of Lake Superior, and this is what the vegetation looks like. It is a dying birch forest with very little new tree species coming up in the understory. And so this is the platform upon which I have been doing collaborations with the Nature Conservancy, Minnesota Department of Natural Resources, to test what it is that we should be planting here. And so here's just some of the planning partners and some of the implementation partners from the Nature Conservancy, and also my own graduate students. So we started this project maybe 7 years ago now, and we used existing data to help inform our plan, like what should we plant. Minnesota is a hot spot for for climate change. Already, there's been 1.5 to 2 degrees Celsius change in our area. And so this corresponds to about a 240 kilometer shift in the climate that was in southern Minnesota, moving up into our region along the edge of Lake Superior. And if you look at the vegetation of the state, what you notice is that 240 kilometers south, it's more mixed, deciduous, as opposed to boreal species in the more northern climate. So this suggests that maybe we need to be using more southern species in addition to seed sources if we're thinking about planting for the future. We also use climate models to inform our plan. So here's a model by one of my colleagues that shows boreal species are really predicted to decline and temperate species are predicted to increase. And so we focused on white pine, bur oak, and red oak because red maple's already getting into these areas and basswood seeds just simply weren't available. And we use state policy to inform our planning. So right now, these are the seed zones, seed planting zones of the Minnesota Department of Natural Resources. And their rules are that if you want to plant in seed zone 104, then you need to obtain your plant material from seed zone 104 and plant it back into the same area. So the idea that local is best. But what we wanted to test was maybe it's better to go a little bit further south and plant into northern environments. And so this is the experiment that we did. We planted 110,000 trees into 2,000 logged acres in like 2012, and some subset were in a formal design so we could go back and identify them and measure them really careful, which we've been doing up until and continuing right now. And our major hypothesis was that plants from a more southern zone, ecotype if you like, 105, would grow better when planted in a northern environment because the climate change has already happened. And so this was a cool project because we achieved the applied goals, which were to do a big restoration project. And we also achieved academic goals by collecting data, analyzing it, and you can read more details about it in this paper, but I'll tell you a little bit of the results. Some of our trees were planted containerized, some of them were planted bare root. This is what the sites look like. We tagged the 5,000 that we tracked carefully, and they were protected from deer browse. And then some were in a formal design so that we could go back and actually locate each specific tree. And then we tracked their survival, their traits, in the environment that they lived in. We looked at how fast they grew, both in terms of height and stem diameter, how thick their leaves are. I mean, think of a succulent, right, with a big thick leaf. That promotes water conservation. And we also monitored the timing of events or phenology. So like in the spring, we went out there and looked at when the buds burst for the pine and also for the oaks. And then for the oaks, we also looked at fall phenology, like when did the leaves start changing? When did they fall off? And so for environmental attributes, we looked at what other plants were on the ground, what the soil was like, and also how much light individual trees were getting. So after 4 years, do seedlings planted one zone further to the north do better? So I'm just going to show you oak data today. And the first thing we're going to look at is survival. So first let me say that there is high survival throughout this experiment. These trees that are right on the edge of their northern distribution did very well. As you can see, survival rates are all over 90%. And for both species, the more southern population, 105, did better than its northern counterpart in terms of survival. However, that was only statistically significant for red oak. That's what that little star means. And just kind of same trend for bur oak, but not scientifically acceptable to say they're different. So how about the timing of events? Well, here is bud burst date on the top, and you can see the more northern, the more southern population in the dots here. Both species burst their leaves out later, and they also dropped their leaves later. And in combination, if you look at the timing between the day the first leaf burst and the timing that the leaves dropped, we can look at how many days of active photosynthesis was happening. And you can see that the southern population of both of these species retain their leaves for longer, which perhaps means that they're able to have greater transpiration and carbon gain, photosynthesis, over the growing season. How about for leaf thickness? Well, here you can see for both southern populations of both species, the southern population had thicker leaves, and that is usually associated with greater water use efficiency. And I just want to show you a fun picture from a drone that was taken this summer of one of my graduate students who is actually measuring the rate of water loss on these trees by putting this giant chamber over them and actually looking at how much CO2 and water vapor is coming and going out of these plants. Growth rate is a little bit weird. The northern bur oak grew faster above ground, and the southern red oak grew faster above ground, but it still could be adaptive in the way bur oak grows, because this species generally tends to allocate more energy to below ground biomass acquisition, especially in the juvenile years. Here's just a comparison of root to shoot ratio. And so you can see that generally bur oak grows by investing a lot in roots at the beginning, as opposed to red oak, which invests more in above ground biomass. So that below ground, if there's below ground allocation in that way, slower growth of the southern population might actually be advantageous. So do these trait differences even really matter? And we can ask that question by looking at whether or not a trait like leaf thickness impacts survival. In other words, is there a correlation between whether or not a plant survived and whether or not it had thick leaves or early bud burst? And so that's what this table is showing. And so anything that has stars means that it's significant. And so what this is showing, the negative sign means that for bur oak north and south and red oak north, it was really important to burst buds earlier. That's why that sign is negative. Whereas red oak 105 seemed to be doing it just about at the right time. And if you look at leaf senescence, you can see that it didn't make any difference when leaves fell for bur oak, but for red oak, the ones from 105 that dropped later had a big advantage. For all of the populations, having thicker leaves was better. And this resulted, and also trees that had higher growth rates ultimately ended up surviving better. So, should we be doing climate informed restoration now? Well, this study showed that at least with these three tree species, um, the one that each has a distribution near the northern edge of the species range, it might be a good idea. They had high survival, and it also looks like southern trees do better in northern environments, and that selection is favoring plants that are able to keep their leaves out during the longer growing season that we have now, that they have thicker leaves that might help them conserve water in late summer drought, which is becoming more common. And this experiment will continue to provide more evidence of this into the future. But the question that I would like you to leave you with is whether or not we have enough information to do this on a broader scale. And I would say that we should continue to do academic and applied collaboration on many different species, and achieve both the applied and the academic goals while also improving our understanding of how well this approach might actually work. And I would like to thank you very much for your time, and I'd be happy to take questions.
Good morning, everybody. My name is Anna Bucharová, and I am from Germany, from the University of Münster. And I am going to tell you something about production of seed for restoration in Germany. What are the important characteristics affecting selection of seed for restoration in Germany? There is a lot of emphasis to use as local plants as possible, and this has two reasons. One is local adaptation that have been shown to be quite widespread in Europe. And the second one, in intrinsic value of local gene pool. What does this mean? Differentiation between plant populations is not only because of the local adaptation, but also because of historical factors. During last glacial maxima, the plants were basically only here in South Europe, because in the north there was ice. And in the South Europe are so-called refugia. And from these refugia, the plants then recolonized Europe. And of course, plants that come from different refugia, they have different genetic background, and they are less related. And these genetic differences are part of biodiversity. They are perceived as important, and there is a lot of stress on maintaining this diversity. Another factor that is increasingly important is adaptability to novel environment, which basically means increasing genetic diversity. This will be more and more important with ongoing climate change. And the market of seeds for restoration in Germany is regulated by law and is mandated to use local or regional seed. How does it work in Germany? Germany is divided in 22 seed transfer zones. That's the thin lines in the map left. And seed transfer is allowed only within these seed zones. However, the seeds are mostly produced, and it wouldn't be profitable for the farmers to produce seeds only for these seed zones within these seed zones. For that reason, there were delimited so-called production areas that include multiple seed zones. That's the thick lines in the map. And the farmer can produce seeds on one farm within these production areas for all the seed zones that are in the production area. Important is that the cultivation for the different seed zones are far apart, that there is no gene flow between them. How the production works. Seeds are collected from at least 5 large natural populations in the region. Sticking to the region, to the seed transfer zone, preserves local adaptation and the genetic pattern due to historic factors. And mixing the seeds enhances genetic diversity, especially in comparison with a single population source. These F0 seeds are then sown into the field, and they give rise of the F1 cultivation. That produces F1 seeds. These F1 seeds are sometimes marketed, but since they are usually not so much, they are mostly used for establishing F2 cultivation. And these seeds of this F2 cultivation, they are then available for restoration. This repeated establishment of cultivation can go up to 5 generations and then they stop. And the reason is that the production process could alter the genetic structure of the population because of unintended selection or genetic drift. The system works quite well. There are 2 large companies plus multiple small ones, but the large companies, they provide seeds for the whole Germany for nearly all seed transfer zones because they have seed producing farms in all the production areas. You just order the seed on the internet and they come by post. For most seed transfer zones, you have like 10 to 20 species available, but for some of them, you have up to 50. And this is how such production field looks like. You see hectares and hectares of wildflowers that grow in monoculture. The condition in cultivation is pretty much different from the conditions in the wild. In the photo on the left up, you can see the wild populations, and on the photo down, you see the cultivation. The plants are growing in the monoculture, they are watered, there is often applied pesticide. And there is general fear that actually this cultivation condition could impose selection, and it could change the genetic composition, the genetic properties of the cultivated population. Additionally, when only subset of the seed from the previous generation is used for establishing the next generation, this could be actually a repeated bottleneck, and there could be genetic drift and loss of genetic diversity. This was so far feared, but actually never tested. So we decided to test it. And we obtained 15 species from 2 seed producers. And the different generations, the F0, which they had in archive, that was really the seeds that they used for establishing the cultivation, up to the maximum cultivated generation. In some species, it was F2. In some species, it was up to F5. And we took these seeds, put them to common garden, took leaves, and did DNA analysis, specifically genotypic by sequencing, to look how actually whether and how much is the F0 differentiated from the later generation. The work was done by my PhD student Malte Conradi. What he found out here, this is about genetic differentiation. On the x-axis are the consecutive generations in cultivation for each species, and on the y-axis is the genetic difference towards the wild plants, towards the wild populations. And you can see that the first cultivated generation is less differentiated from the wild plants than the later generation and the later generation and so on. It is actually a linear relationship. So in fact, the longer the plants, the population is in cultivation, the more genetically different is the cultivated generation from the original wild collected seeds. This sounds quite dramatic. The cultivation has effect on genetic composition of the populations, and the effect is the larger, the longer the population is cultivated. But let's put this to the context. The graph on the right side shows the maximum differentiation, the maximum FST, which we observed between wild and cultivated generation. Between wild and the latest cultivated generations. For some species it was F2, for some species it was F5. So this corresponds to this part, actually the highest effect. However, when we put it to the context and look how plants are differentiated between the region, this is this box plot, we see that the genetic differentiation due to the cultivation is minimal. It is basically negligible in comparison to genetic differentiation of plants between regions. So it means, okay, the cultivation has effect, but the effect is minor. So this was genetic differentiation. Now let's have a look on genetic diversity. On the x-axis are the different generations from wild plants to the last cultivated generation. And on the y-axis is genetic diversity. And you can see that actually the cultivated generations have higher genetic diversity than the wild collected plants. We are still not certain why we see this pattern. It can be because of relaxed selection or admixture in the cultivated field. So in summary, the effect of cultivation was rather minor, and the fears which were there that there is reduction of genetic diversity and big change of genetic composition, we didn't find this in our results. One thing what we should probably point out is that the German farmers are aware of this, and they are trying to basically prevent selection. They are definitely not imposing artificial selection in the way that they try to select the best plants. They really try to avoid any effects. So these have been differences on the level of DNA. But we also looked on the phenotype, and the phenotypic differences are maybe more important because with phenotype the plant reacts with its environment. And what we did, we took the same seeds and planted them side by side to a common garden. And since the plants in the common garden face the very same environmental conditions, the differences which we would see are given by the genotype. Let me show first results. This is about phenotype and specifically about biomass which the plants produce. We let them grow and then we cut them and dried the biomass and weighted it. How to actually interpret these graphs? These graphs are called forest plots, and they show the differences. Here on the y-axis are the different species and producer combinations. So these are the different species. And here on the x-axis is the effect size divided by mean. And basically it means when the point is to the right, the plants get larger during cultivation in proportion, and the values are proportions. When the point is on the left side, plants are getting smaller during cultivation. They produce less biomass. The red points indicate significant results. And we can see that actually in most species there was no significant effect. That's the black points. But in some species there was significant effect, and it was mostly in the direction that the plants are getting larger. So it means that the plants from the later cultivated generations were larger than the plants from the earlier cultivated generations or from the wild seeds. This is different trait, onset of flowering, when the plant started to flower. Again, we see forest plot. On the y-axis are different species, on the x-axis is the effect size, and in this time it is in days. And we see in vast majority of the species there is no effect. But in two species there is significant effect, and in one species, here in Lichnisphos cuculli, the effect is quite strong. For one cultivated generation, the plant starts to flower 1 week earlier. When we have 3 cultivated generations, the cultivated plants start to flower 3 weeks earlier than the wild plants. And this is a huge effect. How this could happen? In natural population, there is variability in flowering. some plants flower earlier, some plants flower later. And all of them manage to produce seeds because the seeds eventually fall down on the ground. But in cultivation, important is which seeds are harvested. And when the farmer has the feeling that the most plants produce seeds, they harvest the plants, they harvest them so that they mow them and put them to the threshing machine and the seeds are threshed. But this means that actually the plants that produce the seeds later can't produce seeds, they don't get to the next generation. And with this, the whole mean of the population, the mean flowering time of the population shifts to earlier time because the later plants are missing. And this is repeated over multiple generations. So that's how this large effect could actually happen. The reason for all this effort cultivating regional plants and so on is because we assume that the plants are regionally or locally adapted, and we want to maintain this regional adaptation and genetic differentiation. But is this actually true? Are the plants that are cultivated still regionally adapted and genetically differentiated? To answer this question, we established a common garden experiment where we took plants from 8 origins. Here we took the seed transfer zone as the production area as seed transfer zone because we did it in 2013. In that time, the availability of seed was limited. So here we have 8 seed transfer zones that are actually now the production areas. And we took 7 species from these regions and planted them to 4 common gardens distributed across Germany. That's the green squares. And we tested whether the plants are regionally adapted. That means whether the plants from the region grow better than the plants from other regions. And we found out that it is indeed true. Here, this is again forest plot that shows regional versus foreign. Here is the local advantage. And we measure the fitness or the performance of plants as in 2 measures, biomass and number of inflorescences. And we can see that the plants, the regional plants, produce on average nearly 10% more biomass than the plants from other regions. And this effect is significant. And also nearly 10% more inflorescences, but in inflorescences there was more variability and the effect was not significant. So we can say yes, the regional plants really grow better than the plants from other regions, despite this cultivation process. As the regions were quite large, we also measured the localness also by other parameters. In this case, as geographical distance between the origin of the source population, the original source population, and the common garden. And we can see that the further away the plants are sourced, the larger is the geographic distance between the garden and the source population, the worse the plants grow. And when we go on average across all the species and all the gardens, when we go 100 kilometers away, we lose 2% of biomass. It is significant for biomass. For inflorescences, there is no effect. And we did the same exercise with climatic distance, with the temperature difference between the source and the garden. And here we see significant effect on the number of inflorescences. And the less similar the climate was between the source and the garden, the worse the performance, and it was significant for the inflorescences. And with each degree of Celsius difference, the plants produce 5% less inflorescences. The year when we ran this experiment was rather hot. In that time, it was the second hottest year on record. So this allowed me to test the idea of assisted migration. what I did, I selected from the data set only data on plants that are either regional, come from the same region, or on plants that come from regions that are warmer, that are 1.5 to 2 degrees warmer than the local provenance, because that was the temperature that actually the plants faced in the common garden. And I expected that actually, because it was so hot, the plants from the warm areas will perform better than the regional plants because they should be warm adapted, so they should have advantage. And I was surprised that this was actually not true. In most cases, there was no difference, and the only significant difference was in Lichnis, that's here, and it was negative. That means that despite the hot year, the regional plants perform better than the plants that should be better because they come from warmer area. To sum it up, Germany applies a mixed and match system where seeds from multiple populations from region are mixed, and the aim is to maintain regional adaptation and increase genetic diversity. Cultivation has minor effect on majority of the plants, but exception exists. There are few plants where the effect is large. And the produced plants are regionally adapted and perform well even under extreme climate, also here. And it gives me hope that this system might be fit for climate change. And with this, I would like to thank to Pate for invitation, DFG for funding, and you for attention. Thank you.
We're going to ask a few questions just very quickly.
Thank you. Daniel Carter has had a good question just now in the chat. He is asking, what are the life histories of the species in these experiments? And are they annual, short-lived perennials, or long-lived? And does this make a difference in what demographic transitions are associated with traits? In the meantime, we really highly recommend Dr. Bucharova's paper on mixing and matching source populations in Germany. It's fantastic, and Becky included a link in the chat. Oh, we can hear you now.
Okay, excellent. Yeah, the plants were mostly perennials, but short-lived perennials, and they flowered in the first year of the experiment.
Wonderful. And then, so why do you think your results were different than of herbaceous plants from Dr. Ederson's results of woody plants from the previous presentation?
Yeah, I did quite some search in literature on assisted migration of this idea of the assisted gene flow from basically planting the southern populations more to the north. And it seems that it is highly system specific. And it doesn't look like it doesn't seem to be a silver bullet, which basically solves everything.
But that's what we need.
I think it will really be system specific. But there is one more thing, and that's the habitat. We are dealing with semi-natural grasslands, which are actually grasslands that were made by humans. These European meadows, they are very different from North American prairies. They are basically a result of deforestation. But this deforestation goes back 5,000 years ago, because there was a so long history of the European, in the Europe of humans. So the humans were actually natural force in this respect. But it means that without humans, this habitat wouldn't be in balance with the climate. There would be forest. And this might also affect the results.
Thank you. And one quick question, last one for you is, how your research, what results on the ground has it had, and have approaches changed in Germany because of what you have shown?
Actually, we were testing the current system, and we just confirmed that the system works, which was actually a lucky situation. What is now in Germany is a bit problem that they are really trying to go as local possible, and as basically I'm afraid that now the practitioners or the policymakers, they are so focused on sourcing locally that they have potential to actually kill this system. because with the stress on very, very regional, and that actually this seed that is produced as a regional seed and sold, that it is not actually not local enough. And I don't know what will happen there, which would be really a pity if they kill it.
Right. Well, thank you so much for your presentation, for answering the questions, for tuning in all the way from Germany.
Thank you for your
Thank
attention.
you. And the presentation will be posted at a later date on the website. So those who've had some technical issues viewing it will be able to view it again and again, as I plan to do, to continue learning. Thanks again. And we will now proceed with the following presentations. And questions that were asked for Dr. Ederson will be addressed after the next presentation.
We will now hear our first round of lightning talks with Andrew Bauer of the U.S. Forest Service, Matt Olson, Lake County Forest Preserve District, and Bill Kleeman from the Nature Conservancy.
My name is Andy Bauer, and I'm a forest geneticist with the U.S. Forest Service based in Olympia, Washington. I'd like to thank the organizers for their invitation to give this talk, and I'm going to be speaking about seed planning, management, and deployment for future climates in the Pacific Northwest region of the Forest Service. And this is the states of Oregon and Washington. Our approach in Region 6 is that we're trying to create a new framework for seed movement that's based on climate rather than geography. The issue is that the Region 6 seed zones have become dogma, but these zones have rigid boundaries and assume a static climate when we know that climate is changing. So what we're trying to do is to transition decision makers to a different way of thinking. One key concept is the decoupling of the seed collection and seed deployment area constructs. In the past, seed was collected from a designated area, seed zone, and it was deployed back into only that same seed zone to minimize the risk of maladaptation. But plants are adapted to the prevailing climate, not to the specific latitude, longitude, and elevation of the site where they came from. Climates are changing and will continue to change in the future. So the seed zone or collection area which is static, while the appropriate deployment area for the seed is a moving target. So these are not the same anymore. In Region 6, we've been working with the seed lot selection tool that was developed by the Pacific Northwest Research Station and Oregon State University to enhance our seed planning, collection, and deployment decisions. The seed lot selection tool is intended for forest tree species, but there is a related tool, the Climate Smart Restoration Tool, that is intended for restoration species. We use the seed lot selection tool to characterize the climate adaptation profile of our seed lot inventory for a whole suite of climate variables. And to revise our seed deployment guidance, we're looking at the mid-century or 2041 to 2070 time span. And winter temperature is one of the key factors driving local adaptation in forest trees. So we used a plus or minus 2 degrees Celsius threshold as our primary factor. And by planning for mid-century, we can select seed lots that match the winter temperature for that time period, but they're still within that 2-degree window both now and at the end of century. So this minimizes the risk of frost damage at the critical seedling and sapling stage, and also provides flexibility as a bet hedging measure in the long term. To complete the linkage to our seed inventory system, this climate data was fed into a spreadsheet that makes the calculations of transfer distances. So then this table can be sorted by the time period and the climate variable of interest to help in selecting seed lots that could be used in reforestation. For example, here, for a selected planting site, this seed lot, which I've highlighted in yellow, is a great match for mid-century, but it's also within that threshold of plus or minus 2 degrees for winter temperature, both in the current time span and also at the end of the century. Now, there's some challenges with this, including the fact that for many of our older seed lots, they were bulked over larger areas and climatic gradients. So it's difficult to characterize their climate profile exactly. So we're using the climatic center of the zone as an average. Another challenge is that our inventory is quite large with well over 1,000 seed lots. So we're having to prioritize this work. We've started with Douglas fir and ponderosa pine, as these are the most commonly used species for reforestation. And then other species of conifers on our inventory will follow. And looking to the future, our guidance for new collection is to bulk seedlots over a narrower climatic range and record detailed location information so that the climate profile of the seedlot can be determined more precisely to aid in deploying it to the best location. Thank you very much.
Hello everyone, my name is Matt Olson. I'm a restoration ecologist with the Lake County Forest Preserve District. I'll be talking a bit about the Forest Preserve's historic seed sourcing, our current practices, and our native seed nursery. Prior to the 1990s, seed installations were very small and mostly involved locally collected seed from staff and volunteers. In this prehistoric era before GIS, seeding event records are somewhat incomplete, but it appears that a lot of seed was likely sourced from within Lake County or the Chicago region. Seed was collected and moved within a site or between preserves with the same or similar habitats, and sometimes seed was shared amongst other districts or agencies. Throughout the 1990s, seed purchases were rare, but when they occurred, seed was specified to within a 200-mile radius of Lake County. As you can see, this 200-mile radius extends beyond Illinois and into Wisconsin, Michigan, Indiana, and Iowa, and even touches portions of Minnesota and Ohio. Referendums of the 1990s and early 2000s provided funding for the acquisition of new preserves, essentially doubling the total acreage owned by the district. This funding also allowed us to initiate multiple large restoration projects concurrently, including the retirement and restoration of nearly 3,900 acres of agricultural land. These projects expanded our need for larger quantities of seed. For us, 2012 was the dawn of a new era for seed sourcing, as staff created new specifications for seed purchases. This effort compiled input from all staff ecologists, but was really led by Kelly Schultz, the district stewardship ecologist, who was our native plant nursery coordinator at that time. The result of this effort was a tiered system. In making these tiers, staff considered things like the natural divisions of Illinois, glacial history, dominant soils, ecoregions, and other research on this topic. Other research including climate change. I think we have all seen these diagrams or something similar, which shows how our climate is predicted to change by the end of the century. If climate change continues as predicted, Lake County's climate will be similar to the current climate of Texas. Therefore, species and their ranges will likely shift northward. These factors led us to adopt a tiered system. Lake County is indicated on the map by the green dot, and the circle shown is a 200-mile radius from Lake County. Tier 1 is made up of the areas with similar soils and glacial history to Lake County, and those with current or predicted future climate similarities. These tiers are specified by county in each state. Seed sourced from Tier 1 can be used within any preserve or project location. If a species is not available from Tier 1 provenance, ecologists have the option to purchase the species from Tier 2 if available. Seed originating from Tier 2 may be acceptable for certain preserves and projects, depending on the species in question and its life history. We consider things like how the plant is pollinated, the rarity of the species in Lake County and in our region, and whether the species is habitat restricted. Each restoration ecologist ultimately makes the judgment call on whether or not to purchase each Tier 2 species on a site-by-site or project-by-project basis. Seed with a provenance outside of these tiers is generally not considered. In our bids, we state a preference for Tier 1 seed. Therefore, we are required to purchase the seed from a Tier 1 source, even if a Tier 2 vendor can supply the species at a lower cost. This is directly stated in our seed specifications, as is the tier map and list of counties. So you might be wondering, how does this affect pricing and total project costs? We do not perform a cost analysis annually, but we have explored this at various times. Generally speaking, we are able to source about 85% of the seed purchases from Tier 1, and the remaining species come from Tier 2. Seed pricing and availability change every year, and not every vendor provides a bid each year, so it's difficult to make definitive statements about the quote-unquote cost of implementing this tiered system. For our most recent seed bid, I looked at all the species that I could compare Tier 1 versus Tier 2 pricing. For example, 2 vendors supplied prices for the same species, but from different provenances. Of these 49 species, the Tier 1 vendor supplied the lowest bid price on 32 items. So for the most part, the lowest bid price generally comes from Tier 1 provenances. By stating a preference for Tier 1 species, we are not always purchasing seed at the lowest price. This preference means that our seed costs were approximately 4% higher than if we had only purchased from the low bidder, regardless of the tiered system. In other words, enacting this tier system increases our seed cost totals by about 4%, but we do not consider this a significant enough cost for attempting to preserve local genotypes and account for future climate change, nor do we consider saving 4% a significant enough reason not to consider these things. This tier system is not just for seed. We use this system every time we're purchasing any plant material, including all plants and wetland plugs, as well as trees and shrubs. At the same time the tier system was enacted, we also created a do not buy list. This is simply a list of each native species in the plants of the Chicago region with recommendations for seed purchases, and this is our attempt to preserve local genetics. So some reasons why a species might be on our do not buy list: if the species was not historically present in Lake County, we don't buy it. If a species is rare or uncommon, or is a threatened endangered species. If a species only occurs in a rare or specialized habitat, such as what's only found in bogs, fens, or ravines, for example, then we may not purchase it. Perhaps the species is part of our current in-house production at our nursery facility, or the species is easy to collect in sufficient quantities from wild populations to meet our annual needs. Lastly, species with restricted pollinators or limited dispersal, such as spring ephemerals, offer a good opportunity to preserve local genotypes. Additionally, we have some sites designated as do not buy sites. These are preserves or portions of preserves which we believe host high-quality remnant populations of numerous species, places that have not experienced seed introductions from purchased material in the past. The intention is to keep these sites pure, whereby only locally collected seed can be used at these sites. The Lake County Forest Preserve District operates a native seed nursery encompassing approximately 4.5 acres at our Rollins Savannah Forest Preserve located in central Lake County. The purpose of the nursery is to produce seed and plants that may not be commercially available, produce local genotype seed, and decrease our seed expenditures. This allows us to reliably and sustainably obtain locally sourced material. In summary, for closing, the Lake County Forest Preserve District does not have a written or formal seed sourcing policy. We do have some standard operating procedures and specifications in place to help preserve local genotypes, specifically of rare plants, and we employ a tiered system when purchasing seed to limit seed purchases to within a 200-mile radius of Lake County, but this system offers a preference to plant material from the southern portion of this extent to account for predicted climate change. We also operate our own native plant nursery as a seed source to help increase the quantity and availability of rare, expensive, and non-commercially available species. And lastly, I just want to say that we are always looking to improve our seed sourcing procedures and strategies, so I look forward to what other people have to share. Thank you.
Hello there, colleagues. Bill Kleiman here with the Nature Conservancy at Nachusa Grasslands. Happy to be presenting to you in these strange COVID times. First off, where are we? Nachusa Grasslands is in north central Illinois, about 2 hours west of Chicago, about an hour south of Wisconsin, hour and a half to get to the Mississippi River. So we know that if you want to have good habitat restorations, you need a lot of weight of seed, you need a lot of diversity of seed, and if you get a diversity of plants growing in your restorations, you're going to have a diversity of animals, and that diversity, I'm making the wild assumption, is going to be good for resilience in a changed future of climate. So how far afield should we go to get that seed? Some would say 0 miles. If you have a cornfield you're going to let go fallow, they would literally let it go fallow. They would not move seed into it. They would assume the seed is going to blow in or be walked in by wildlife from adjacent areas. I don't agree with this thinking. I don't think it works. I think you just get a funky fallow field, but that philosophy's out there. Okay. Then there's people that are very cautious. They would move seed a 1/4 mile, a 1/2 mile, from this remnant right next door to that cornfield you're going to try and create prairie in. Okay. Then there's folks, I know an agency that chose 15 miles because all of their holdings fit within that distance and they thought that worked for them. Okay. There are some folks who'd say 100 miles is good or 200 miles is good. You would think with the landscape we used to have that was connected, genetic flow moved all across the landscape with wind and wildlife, and it makes sense to me. So at the Chusa Grasslands, we've chosen 50 miles. 50 miles was chosen way back before me. It's like it was big, not too big, just right. And 50 miles is a huge area. Typically, we pick within about a 2-minute drive. So we're not wasting time driving to a site to pick seed. We're on it. And we can bring in a huge amount of seed when you have a good place to pick from. A lot, thousands of pounds from a couple hundred species. An amazing quantity of seed we can get right nearby. And if you're going to travel 15, 20 minutes, you might do that, but there's a loss in productivity as you travel there and travel back. We've also had some experiences in the past that made us hesitate on going far for seed. One time we ordered a native Lespedeza, and what came in the seed mix was an invasive Lespedeza. And we didn't notice it till several years had gone by, and the plant is now on our invasive species list for the preserve. Thank you very much. We also, one year on another order, ordered pale purple coneflower, and what came in was purple coneflower. And now we're going around trying to spray that out where it popped up in several places. Another seed order was an annual bloomers that came in that we didn't order. So these kind of things made me hesitate, at least, for going afar for seed. We sort of became quite cautious about where we would pick seed and how far we would move it. But the 50 miles hasn't been a logistical restriction. We went 70, 80 miles for this one. I remember Todd Bittner way back then wrote a plan for introduction of this rare orchid, the Eastern Prairie Fringed Orchid. We got the plan approved. We got the seed from Lake County Forest Preserves. Thank you, folks. And after about 6, 7, 8 years, a few plants showed up. Chris Hauser, volunteer steward with us, he was also with King County Forest Preserves, was hand pollinating these orchids. And then we got more and more of them, and before you know it, there are hundreds of them, and now we have a stable population, and the hawk moths do their job of pollinating. So going far afield for cool things, rare things, I wouldn't hesitate. So it's nice to know that the diversity we love in a natural area in the plant life also is going to support the animals above it. And some rare species will struggle, and we should work to support those rare species. Other species will thrive. So besides seed, oh so important as it is, we need vigorous fire programs. We need that repeated application of prescribed fire to set the brush back and keep our landscapes healthy. For invasive weeds, we need to create efficient strategies that limit invasive plants. We need to understand what exotic plants are tolerable to live with. And for those that we can't win, what strategies do we have to keep them from going everywhere? So we need to watch and care for our rare species. An example for us at Nachusa Grasslands is Blanding's turtle. We had very few of these turtles when we started doing a monitoring program with Rich King of Northern Illinois University, Tom Anton, and Dave Mauger. For years, we were tracking the Blanding's turtle, and we realized we had very few of them, maybe a dozen. And we saw, I think one time we saw a young Blanding's turtle. So we started protecting nests, and then last year we started taking the eggs to an incubator and doing a Head Start program, working with a couple of partners, DuPage County Forest Preserves, Lake County Forest Preserves, and Richardson's Wildlife Foundation. Thank you, folks. And we release these Blandings here. Our population is now on a, looks good. It looks exciting for us, and we're really happy about that. So we need to protect habitat now for the climate future that's coming. And we could use federal matching funds to help us take our nonprofit dollars and make them do more work. We need to connect our natural areas with wide wildlife corridors. We need to improve the design of our preserves to make them easier to do stewardship, particularly prescribed fire. Here's a design, and then the next slide shows a little simpler design, better for stewardship. So in the future, the money, the funds we need in the future, they're going to be at least what we need now, don't you think? So at Nachusa Grasslands, we supported the start of a Friends group. That Friends group is partnering with the Nature Conservancy to create endowments to keep the project going into the future. Our volunteer stewards program is vigorous and will continue to be so. And then we're working hard the last few years to increase our visitor services so visitors can come out and find the place, find the trailheads, try and understand what we're doing, have a good time, and hopefully changing hearts and minds and loving conservation. So seed is the foundation of habitat restoration. A high diversity restoration, carefully stewarded, produces climate resilience. That's my main pitch. Thank you for your time. Be well.
Now we're going to hear from Jennifer, who's going to talk to us about her experiences with sourcing seed in Arkansas.
Hey everyone, I'm Jennifer Ogle. I'm coordinator of the Arkansas Native Seed Program and collections manager at the University of Arkansas Herbarium. I want to thank the workshop organizers for inviting me to talk to you today about the program we're working on in Arkansas. I'm happy to share this information and to highlight partnerships that we've formed to develop a native seed industry from local plant sources in the state. Our goal is to facilitate the establishment of a native seed industry in Arkansas by developing eco-regional sources of locally adapted native seed for large-scale habitat restoration and revegetation projects in Arkansas. Now, the focus of my talk is going to be primarily the Audubon Arkansas Native Project, which started in 2010, as well as the umbrella project we formed in 2016, which is the Arkansas Native Seed Program. But I do also want to spend a minute to mention that while these programs have been in existence for several years and working statewide to provide native seed sources, local seed sources, some of the partners have been working for much longer, 30 years in fact, to educate agencies working in the state about the importance of using locally sourced plant materials for revegetation and habitat restoration projects, particularly when those projects are situated near a remnant natural area. The main focus of this program is grasslands. There are several reasons for that. One is that they were historically very extensive throughout much of the state. I realize Arkansas isn't known as a prairie state, but General Land Office surveyors came through in the early 1800s and they mapped 750,000 acres of just tallgrass prairie in Arkansas. And we know that they missed quite a lot as they were coming through. The surveyors only mapped what they saw, and some of the surveyors didn't record when they did see prairie. And so the Arkansas Natural Heritage Commission estimates that that number might be twice what was reported. So 1 to 1.5 million acres of just open grasslands, such as tallgrass prairies, glades, and other open habitats. That doesn't include savanna, that number, which was also extremely extensive through Arkansas. So these grasslands were common historically, and they're currently our most critically imperiled and biologically diverse plant communities. And there's a big push toward providing habitat for northern bobwhite and other grassland birds, as well as pollinators in the state. So that's another reason we focus on grasslands, because most of the native seed being brought in are grassland species, and they're being used for that purpose. And so the native seed program wants to replace that non-local seed with locally sourced native seed from our own grasslands. So like I said, I'm going to focus primarily on the Audubon Arkansas Native Project. Now, NATIVE is an acronym for Native Agriculture to Invigorate Ecosystems. And I mentioned that we were working before Audubon got started. There was some seed production being done in eastern Arkansas in the Grand Prairie region. Audubon took over that project, and then they expanded it statewide. And they did this through a couple funding mechanisms. They applied for USDA Conservation Innovation Grants, as well as a 2501 program grant. Now, both of those grants enabled them to work with new farmers, with socially disadvantaged farmers, and military veteran farmers. So the scope of people they could work with was quite large when you take in those three groups. They've also received funding from the Arkansas NRCS grant. And then additionally, they have support from partner agencies. Arkansas Natural Heritage Commission provides support in the form of staff time. Roundstone Native Seed provides match in the form of reduced cost for their plant materials that we send to them and that they process for us. And then the Nature Conservancy has provided funding as well. And then we have 16 partners altogether, and each partner plays a role in this project. And I'll highlight some of those roles as I continue talking to you about this project. Let's go step by step through the Native project and what it takes to make this program work. At the very foundation of this whole program is first site selection and wild seed collection. So we have to identify appropriate sites for collection, and the primary partners who do that are the Arkansas Natural Heritage Commission. They provide information on what sites are remnant grasslands, what was here, what was not planted, what's actually local native. And their natural areas are the primary areas we collect seed from, because we know that those areas are natural and native, and they're also quite biodiverse. They manage their natural areas for maximum diversity. And so that's how ANHC contributes to the Native Project in a big way, is they provide that type of information. But they also send their botanists and ecologists out during collection events when volunteers go out to the natural areas and collect. They're at the events, and if they can't be at the events, they provide real-time identification support. People can call or text message pictures. They send pictures. We use iNaturalist as a tool. Botanists from ANHC will go out and tag populations of plants, and then the volunteers can use iNaturalist to relocate those plants when they're in the field. So we don't just use natural areas, we use other remnant populations of target natives as well for seed collection, particularly power line rights-of-way and roadsides. And this is where our partnership with Arkansas Highway Department is so important. For one of the reasons it's important is they can verify which of their roadside populations were planted. They keep really good records of their native plantings, and so we avoid those areas. And there are some times when we just can't be sure, and so we avoid those areas if we don't know for certain that those populations are natural. And so after the sites are verified, and then we send volunteers out to do the actual collection. Most of the collection is done by hand, especially when it comes to wildflower seed. And one of our other partners, the Arkansas Master Naturalists, have been instrumental in supporting this part of the project. We're fortunate in Arkansas to have a very large volunteer base of conservation-minded people. We have hundreds of Master Naturalists in 6 chapters throughout the state, and they attend most of our collection events and help us collect seed when we're out there. We also engage with garden clubs. The Arkansas Native Plant Society gets involved. And then we post all of our collection events to social media, and so of course just other individuals, unaffiliated volunteers will come out and help us collect seed. We also do some machine collection of wild seed. Audubon, Arkansas owns a flail vac and a tractor, and they harvest mostly the taller native warm season grasses with their flail vac. But then they also recently acquired a push combine, which is this machine on the left that you can see, this aluminum machine, and it's a very effective and inexpensive tool for collecting both wildflowers and grasses. And they can take these into their farms, into their agricultural rows that they've planted, and harvest that way. But they can also take them into remnant prairies and glades with favorable terrain and collect that way. This machine cost only, I think, $3,500, and so it's a pretty inexpensive an effective tool for an organization that's just getting started. And the next step in the process is processing of the seed that the volunteers have collected in the field. The seed is stored at a local facility provided by another one of our partners, Central Arkansas Water, until the fall harvest is complete. And so that seed sits in this facility. It's protected from the elements. It's stored there into fall and into winter. And then once all the harvest is complete, the seed is sent to Roundstone Native Seed in Kentucky. And they take care of the seed from there. They process and analyze the seed, and then if it's wildflower seed, they grow it into plugs, as is shown on the right-hand picture there. Those are some plugs that are getting ready to be planted on a new farm in central Arkansas. But if it's grass seed, it's cleaned and then it's packaged. Grass seed is not grown into plugs. And then the following fall, late summer, fall, the plant materials are returned to Audubon farmers enrolled in the Native Project. So the next step in the process is cultivation. All of the areas enrolled in the Native Project start out as either fescue fields or maybe some traditional row crop agriculture that are being converted to native seed production. No trees were cut. We don't clear land or trees to plant these fields. So all the fields have to be prepped. Most of the time we're dealing with fescue and or Bermuda in these fields, so those have to be removed completely. We get the fields down to bare soil. Now the farmer is responsible for doing this part, and a lot of times they have equipment, sometimes they don't have equipment. And so Audubon will provide technical and on-the-ground assistance when that's needed with their equipment and their expertise. And so in the case of wildflower plots, those are generally 2 acres in size each. And so each plot has to be prepped to remove the non-natives. Then they install drip irrigation and plastic mulch in rows with a machine they have. And then they plant the plugs with a water wheel planter, which is shown in the picture on the bottom left. So 2 people sit on the back of this water wheel planter, and the machine punches holes through that plastic and into the ground, and then these operators insert the plugs into those holes. And then we have a quality control department that goes in behind them. That's the job I usually do when I'm volunteering to help plant a new farm. And this is the owner of this farm in northwest Arkansas that was planted last year. And she's making sure all of these plugs of swamp milkweed get positioned in the holes correctly. So there are about 11,000 plugs that are planted per acre in these wildflower plots. So with the grass seed, it's very different. The plots are much larger. We usually do 10 or more acres. We try to go for that for grass plots. And so the field is prepped and the seed is planted with a seed drill or broadcast. We have volunteers who help with this process as well as we go through it. And so the year after we plant, we generally can harvest everything that we've planted. Most of the species we target and have in cultivation grow very quickly. They produce flowers and fruit in the first year. And so the grass seed can be harvested with large-scale equipment because the plots are larger, and in some cases, particularly in the Grand Prairie region where rice production is common and soybeans, these farmers we're working with have hundreds of acres in traditional agriculture, and so they have large-scale combines and large hoppers that they can use to harvest the seed. And so that's what they use. But the wildflower seed, being that it's in smaller plots, that's mostly harvested by hand. But we can also harvest it with that push combine I showed you earlier. And this is where we engage with volunteers again, sometimes to help collect that seed for the farmers. And so all the seed is dried at that Central Arkansas water facility and bagged up, and then it's sent back to Roundstone after the fall harvest is complete. And from there, Roundstone has it until it's sold. So they process, analyze, package, and market the seed. And farmers have worked out an agreement with Roundstone where they receive a percentage of profits from the seed sales. I forgot to mention also that the farmers receive a stipend through the grant, that the USDA grant that Audubon has. I think it's $350 per acre per year that they're enrolled in the program. So that helps offset some of the costs associated with doing this project. And so this has been a very successful project for Audubon, Arkansas. They have almost 100 acres enrolled right now on 11 farms, and they're working in 4 different ecoregions of the state: the Ozarks, the Arkansas Valley, the Blackland Prairie, and the Grand Prairie. And they have 26 species in cultivation. They have the 4 native warm season grasses of little blue stem, big blue stem, Indian grass, and switchgrass. And then they have 22 perennial wildflowers in cultivation as well. So I'll just show you a few slides real quick about that show what the Native Project farms look like. This is what all of their wildflower plots look like. They're mostly 2 acres each, and they have a variety of species planted. We don't just do one species on a farm. And the reason for that is if one of these new farmers drops out of the program, we don't want to lose all the stock that we have for the species. So they distribute each species, divvy it up amongst each of the farmers. And so this particular farm in the Ozarks near Fayetteville has 13 species on it, including what I just showed you was bee balm. And then in the foreground here we have slender mountain mint, and then just beyond that is pale purple coneflower. We have 2 species of solidago, swamp milkweed, and then maybe you can see in the far off distance here, you can see the stalks of Liatris pycnostachya, or prairie bayfeather, coming up in the distance. And then this is one of our larger farms in the Grand Prairie ecoregion. I mentioned before that they do traditional row crop agriculture in the Grand Prairie on a large scale. And this farmer has 700 acres in corn, soybeans, and rice. If you ignore the grasses in the foreground here, you can see on the left-hand side a field of switchgrass, and then rows of corn, and then to the right you can see, I believe that is big blue stem growing. And so he's doing buffer strips, which is an NRCS practice. So he's incorporating an NRCS practice using our locally sourced seed, which is a great idea. And he's using them as buffer strips between his rows. So that's really the Audubon, Arkansas Native Project in a nutshell. I'd like to spend the remainder of my time talking about the umbrella program that we started in 2016, and that's the Arkansas Native Seed Program. In 2015, as you know, the National Seed Strategy was published, and that was really a motivator for the partners to launch this program in 2016. It provided guidelines for them to follow and really helped them focus, to narrow their focus on what they should be thinking about and how they should develop their own program. So they began meeting quarterly. All the partners would get together quarterly. They had great ideas, but what they found very quickly is no one had time to follow through with those ideas once in between meetings. And so they realized pretty quickly that they needed to hire someone to work on this full time. And so in 2018, they were able to set aside some funding from existing grants to hire a coordinator. And so I was hired to do that in 2018, and I filled that role formally for 2 years. Because of difficulties with hiring new staff at the agency level, they went ahead and hired me as a contractor. So that's how I filled that role, was as a contractor for the Heritage Commission. So one of the things that was done early on when the seed program was launched was they developed seed transfer zones. And the people who worked on this were the Arkansas Natural Heritage Commission, Theo Witzel and Tom Fotai, who together have decades of experience working in Arkansas, and they have a very deep understanding of the geology and plant communities of the state. And so they based their seed transfer zones on the EPA's Level 3 ecoregions of Arkansas. Tom Fotai was involved with the development of that map as well. And so you can see in the center, these are the seed transfer zones. These are the zones they created, and they look very similar to the Level 3 ecoregions. We have 7 Level 3 ecoregions in Arkansas. We reduced it to 6 for the seed transfer zones. And they are based on ecoregions, but they're also based on geology, landform, and plant communities that are shared across adjacent ecoregions. And so here's an example of that, those shared landforms and geology. In the Boston Mountains and the Arkansas Valley Hills, which are in 2 separate regions, these are Level 4 ecoregions, I should say. They were combined because they're almost indistinguishable from each other, and they share similar plant communities through them. So they thought it would be fine to develop one seed transfer zone for that. And these are preliminary. These are based on what we know now, and we acknowledge that these will change. They could change. They should probably change as we fund research on genetic variation across ecoregion and habitats, and other types of research that may need to be done on our plant communities. So our mantra here is data will inform strategy. We've done several other things from 2016 to now, some things I've been involved with. All the items on this list are things I was more deeply involved with. Determining current demand was really important. We wanted to know if native seed industry could be supported in Arkansas. And so the Nature Conservancy helped us a lot with this process, another one of the partners. We developed a form that we sent out to all the state and federal agencies and nonprofits who use seed in the state. And we asked for a 5-year average for what they use. And they came back to us with a 6-year average, so that was great. We got a little more data than we thought we would get. And we found out that about 16,000 acres are covered in seed a year in the state by these agencies and organizations. Now, that's not all native seed, but we're hoping we can tease out that data and see what is native and what's not native. And of the non-native, what can we replace with native species, and down the road with locally sourced native species. So that was an important thing that we can check off our list. We also developed a target species list for each ecoregion in the state. We focused on workhorse species, which is what Audubon is already working on primarily, and we wanted them to have high ecological value to wildlife as well. Another thing we did was we determined what our research needs are. And then I was very involved with writing the strategic plan, and that's the culmination of my 2 years of research as a contractor and collaborating with the partners and visiting with other state and regional programs. I was really helped out by Iowa's Tallgrass Prairie Center, staff there, the Texas Native Seeds Program. I visited with the Pollinator Smart Program in Virginia, and I also visited with some nurseries and seed producers in Missouri. And then we have a list of things that we need to be working on and things that we're just starting to work on with the Native Seed Program. We need to develop demand. We need to train volunteers to collect seed using established protocols. Right now we're taking them into the field and training them on the job, basically. We need to train them ahead of time and follow established protocols for seed collection. And then the two really big things that we need to be doing that we're not doing right now, and I'm sure you noticed as I was talking, that question might have popped up as I was talking about the Native Project. Are we saving seed back? Are we seed banking? Are we developing ecotypes? And the answer to that right now is no, we're not, but we understand that we need to be doing that. And so that's one of the big items within the strategic plan that we wrote, is to increase short and long-term seed storage capacity and also develop sustainable ecotypes of target species. We also acknowledge that we should be giving our seed to national seed banks, portions of our collections to national seed banks. So the reason for this is so that we can move quickly. If there's a rehabilitation project that we need to work on, we can do that quickly if we have seed in storage. And then also, as we modify our seed transfer zones based on research that comes in, and we know we will be able to move quickly to steer in that direction if we need to. And then, of course, we want to develop sustainable ecotypes by collecting from more populations and mixing those as appropriate, so that we can increase genetic diversity of our seed mixes in the hopes that they will be more resilient. So that was a really quick overview of the Arkansas Native Seed Program and Audubon Arkansas's Native Project. If you have any questions or comments that weren't covered during this presentation, please feel free to email me. Don't hesitate to reach out. My email is listed here. It may take me a few days to respond, but I will respond. And if I don't know the answer to your question, I will make sure to forward it to someone who does know the answer. All right. Thanks. See ya.
Thanks, Jennifer. That was great. We do have some questions coming in through the chat function, and Isa is going to present those to you. We're going to ask them of both Julie Ederson and Jennifer. And if there's any questions for any of the lightning speakers, we might have time to answer those too, but I'm not sure because we're definitely running a little behind. But Isa, I hope there were some good questions that came in.
Yeah, so Becky Collins from Cook County Forest Preserves has a question for Julie. She asks, for the Minnesota experiment, did you look at if the southern and northern species are mixing genes? You're on mute. I do this all the time.
Thanks. We have, well, we planted seedlings. They are now about, I don't know, 8 years old or 10 years old. And the very first one produced acorns this summer. And so, you know, if the question is mixing genes, between populations from the south and the north, we don't really have a great answer to that yet. But like other people who have talked about lake effect, we have that issue in Minnesota too around Lake Superior, where the coastal zone, although it's not recognized as a distinct tree zone, is actually really the climate is really very distinct and gets progressively more arid going south or going interior away from the lake. And we have done molecular studies looking at differentiation along that climate gradient only in red oak so far. And actually we pick up very little molecular differentiation going away from the lake. As far as mixing species, there is hybridization with pin oak, and we have noticed that in our red oak samples. So that adds a little bit of complexity to our whatever strategy. I hope that answers the question.
Becky, if you have further questions, feel free to turn on your mic and ask, or put it in the chat. We'll just go on and then we can go back. Jeffrey Zions asks, and I think, Jennifer, you spoke to that a little bit. While most of the talk for sourcing is for climate resilience, focuses on geographic area, how much consideration is given to the difference in soil types in geographically distant areas, particularly in glaciated versus unglaciated, recently speaking, areas?
Well, for Arkansas, we considered soil pH differences quite strongly for our seed transfer zones. And so we have, blackland prairie versus acidic prairie, calcareous glades, acidic glades. So we consider those to be the finer zones that we might do more targeted restoration and collecting and keep those separate at this time. Or maybe down the road, we'll consider those finer zones as we get more proficient at this.
And in the Minnesota study that I described, we did measure soil attributes, and they definitely had an effect on the growth characteristics and other traits of the species, but it didn't overwhelm the signal of climate.
Okay, so we can go to the next question. Carrie Pike asks, for seed orchards or seed beds that will be established for future seed collection, should we maintain weak stunt geotypes to increase genetic diversity or impose selection by culling plants that are runts and possibly maladapted? I love that.
Yeah, we want the runts. We want all those differences to be maintained in our collections. And so we'll never move to all mechanical harvesting wild seed or our mass-produced seed. We're going to have to be really inefficient in that way to keep that genetic diversity that we purposely built into the mixes in the cultivated product. So yeah, definitely we want the runts.
I agree completely with Jennifer. I think that we do not know and we cannot predict what will be the genotype that is favored by selection in the future. And so I think maintaining genetic diversity, even if some plants appear to be runts, I think is the right move. And from having done a lot of natural selection studies in the field, I have a lot of faith that if that runt is maladaptive, it will be eliminated from the population naturally.
And when we send out a summary of today's meeting, we'll try to include some articles that can give future hints or suggestions on how to proceed. There is a great article from the Chicago Botanic Garden on sourcing, cleaning, and collecting seed from the wild for increasing genetic diversity, and goes through like, I think 10 steps, and I think is by Andrea Kramer. So they'll be linked. Jennifer, you have a follow-up from Jacob. He asks, how did you determine the 22 species that you collected and farm? And are there plans to change or expand the species list?
That's a good question. That's what Audubon has in cultivation right now. They're all perennials. All of the materials they have are all perennials. They were somewhat opportunistic as they went into the field. They would, if they saw something that was ready, they would collect it. Also based on our knowledge of what's in those remnants. But our current list, and something I helped work on, is much larger. It has 85 species: annuals, perennials, native warm season grasses, native cool season grasses, more legumes, just a lot more variety. And so we have changed that to be more strategic in what we're collecting so that we can develop mixes of those things.
Great.
And so I should say, the first question, how did we determine the 22 species? We were opportunistic, but they picked all things that are considered workhorse species that would do well, they're abundant, and they perform quickly, and they're harvestable after the first year. Those were the general guidelines.
Okay. And Gary Sullivan of the Wetlands Initiative asks, considering that Illinois is predicted to be moving towards the climate of West Texas, should we be considering the efficiency of sourcing genetic material from further south, and in some cases much further south? I think those are some of those big questions that we're trying to answer here today. Anyone comments?
Well, I'll just say that I think that incremental change or incremental movement is safer. I guess my inclination, the way I like to think about it is actually just kind of restoring the avenues of gene flow that would not have been there without habitat destruction. And those are more incremental shifts across space, of course, depending upon what kind of pollination system there is for the species. But what I really feel like is that there is weak information. There's just not enough information. And we already know that the patterns are going to be species specific. And so I really strongly advocate for the idea of restoration ecologists partnering with academic people who have like the graduate students and other resources to get out there and measure the restorations that they're doing that are climate forward in order to test the efficacy of this approach.
Okay, I think those are, that's all the time we have for questions now. I know we're moving fast, but this is just the first step. We'll continue this discussion. Thank you both. And Patty, take it away. We're gonna proceed with the program.
Thank you. It's time to go to the next breakout session. Our second round of lightning talks features the seed producer's perspective, and we will hear from Jason Fritz with Cardno in Indiana, Andy Starr with the Prairie State Nursery in Illinois, and Corrine Daniels with Taylor Creek Nursery in Wisconsin.
Good morning, everyone. I'd like to share with you a brief look at native seed production from the grower's perspective. My name is Jason Fritz with Cardno Native Plant Nursery. I've been with Cardno since 2001, and I am the Business Development Manager for our nursery. My primary role is to lead the nursery sales and marketing efforts and manage our inside sales staff. I'm also the lead contact for custom seed mix design and oversee the development of standard mixes listed in our catalog. A brief history of our nursery. Nursery started operations in 1994 at our location in Walkerton, Indiana. The nursery facility is located at the northeastern end of Ecoregion 54. Our material originates primarily from counties in northwest Indiana, including ecoregions 54, 55, and 56. The nursery has over 95 acres of row crop seed production. Of that, 69 acres is for upland species, and 26 acres is for wetland seed production. We currently have over 110 species growing on our nursery grounds, which we supplement with off-site collection on leased ground, which provides about 80 additional species. I'll quickly go over our production cycle, which begins in the fall of the prior year, when we review our estimated demand and our current production. We'll decide on species and quantities for future production and start putting that plan into action during the winter months. For on-site production plots, we develop a plug list, reserve the greenhouse space, and start growing those foundation plants. New production plots will be prepared and planted in the spring. Most of the time, plots start to produce large quantities only after 2 to 3 years. Seed collection begins in May and runs through November, the goal being to collect the seed from the plant before it hits the ground. Most cleaning will not start until after the collection season has ended. Cleaning usually extends into February. As lots of seed are cleaned, they will be sent into an AOSA certified lab for independent testing and analysis. For most lots, we will send in a sample for an initial purity and tetrazolium test to get an estimate on viability. If those initial tests come back good, we'll send it in for a full germination test, which could take an additional 3 to 6 weeks depending on the species. Most small lots of rare hand-collected species are too small to send in for a test. How does climate change affect what we do at Cardno? Well, I recommend taking a look at a report issued by the Indiana Climate Change Impacts Assessment entitled Indiana's Agriculture in a Changing Climate. They presented several key findings. The most salient point for us is this. Warmer winters, wetter springs, and hotter summers may result in increasing weed, pest, and disease pressure on Indiana's agricultural production. These indirect influences are complex, difficult to predict, and can be positively or negatively affected by farm management decisions. We are currently seeing those impacts now. Every spring seems to be wetter than the last. At the nursery, it can delay our access to production fields. This can disrupt new production plot installation and early season weed prevention measures. Rainfall on the average seems to be increasing, but there seem to be longer periods of drought. This year especially, we have had to rely on irrigation more than the past. It has run almost every day since the end of June. Weed pressures also continue to increase. In 2011, Palmer amaranth was found in Indiana, and those populations were able to withstand treatments equal to 7 gallons per acre of generic glyphosate. Mare's tail has also become more difficult to control with traditional herbicides. So, what are we doing to solve those problems? In the past 2 years, we have started using more plastic mulch beds. This allows for increased soil moisture retention and decreased herbicide over traditional row cropping. We have also started using more pre-emergent herbicides in our plots. This cuts down on our post-emergent spraying, reducing non-target mortality, and produces a cleaner crop. For bare root wetland plants, we are switching from in-ground wetland production ponds to lined and raised bed production. So, origin genotypes. Currently, we are not shifting the genetic origin of our stock for seed or plant production. Currently, there just isn't any demand from the consumers for more southern stock. We're hesitant to make that switch for several reasons. The largest one is we don't know where the demand will be, and we don't know how far south to go. As you can see from some of these projections, it could be from anywhere from south central Texas for summer temps to eastern seaboard for winter temps. As I mentioned before, a new seed plot will take 3 to 5 years to get to full production capacity. This will also limit the availability of rare, threatened, and endangered species. Importing new genotypes of species could result in hybridization with other native populations. In reality, there are just too many questions that are unanswered for producers to make this shift. It took decades for Midwestern native seed industry to grow to the levels of production that it is at now. If a shift in plant origins is requested, it will take major commitments from all parties, especially the end consumer, to make this a reality. Thank you for your time. If you have any questions or need clarification, feel free to reach out to me at any time.
We have Andy Starr up next.
Hello, my name is Andy. I am one of the owners of the Prairie State Nursery, located in LaSalle County, Illinois. That's me on the right. I'm a landscape architect and have spent the last 15 years in the restoration industry with the Pizzo Group. I currently am employed by the Illinois Department of Transportation as the Roadside Management Specialist for District 3. On the left is my partner, Brian Dose. He is a full-time firefighter with the City of Ottawa and has family farmland that's been in the family for generations, and that's where the farm is located, near Lawston, Illinois. So this is a part-time endeavor for both of us at this point. We both have full-time jobs outside of the farm. So right now, we've expanded 3 times and currently have about 12 acres in production. We supply about 116 different species from 26 different Illinois counties. Mostly prairie species at this point. We do have some wet prairie and woodland edge species, so we have some variety to provide some different mixes. And we're a very good sedge producer, so if you need sedges, we've got several species where we have large quantities of sedges every year. But we are exclusively Illinois ecotype seed. We don't grow anything from anything outside of Illinois. Now real quick, just a few pictures from our farm. These are our production plots. Most of these are from the summer of 2018, so 2 years after seeding. So getting into the meat of the presentation, I was asked, have we seen an increase in source identified seed? I would say there was a push in the early 2000s that to include language and specs that had origin restrictions for seed produced. And what I really seen come out of that is kind of 4 different categories of native seed consumers. That's the scientific community, the restoration community, trend followers, and uninformed consumers. I don't really think that we've seen in our 3 or 4 years of being in business as a nursery a significant increase in the use or the requirement of source identified seed. I think what we have seen is a consistency of it being used by the scientific community. So I would include in that category federal government agencies like the Army Corps of Engineers, many county government, forest preserve districts, organizations that are science focused and have financial resources to pay for good quality source identified seed. That seems to be the native seed consumer category that is really driving and concerned with source identified seed. The other categories I don't really believe are pushing source identified seed unless they're required to do so. So I was also asked the question of, what scale of restorations do I see using source identified seed? And really, I would say the scale of restorations is not as significant as the type of consumer purchasing the seed. So, in general, restorations requiring source identified seed do tend to be a bit larger in scale just because of the type of consumer requiring it. So, Army Corps projects, forest preserve projects that have bigger chunks of land that they're restoring, and those are the scientific community that is pushing or is concerned with source identified seed. I was also asked to talk about the source identified challenges that we've had. So here in Illinois, like I had mentioned, we really have 2 Illinois producers. The other one really offers mostly Northwest Illinois counties. So there's just a lack of supply. And then in us getting started with our nursery, there's a lot of challenges to overcome as far as finding a remnant population you can collect from. And keep in mind, Illinois has 1/10% of its prairies remaining, so there's a lack of remnant systems to collect for, and a lot of those are protected that you can't collect from. So finding a remnant to collect from is a challenge, and then finding a remnant within the array of different ecosystems we need to collect from is a challenge. So, different types of soils, different wetness categories, different shade categories from woods to savannas to prairies. So trying to collect remnant species that's source identified from all of those different categories of sites is very challenging. And once you do get that, you got to get access or permission to collect within those areas, and sometimes you got to compensate the landowner, so that's a cost. And then once you get that, you've got to grow, clean, test, and sell it. So there's quite a process involved, even once you've found the remnant you can collect from. So it is very challenging here in Illinois to start a native seed nursery with source identified seed from Illinois counties. And outside state producers do have some limited quantities of Illinois ecotype seed, but it is, again, fairly limited. A lot of it comes from Minnesota, Wisconsin, Iowa, Indiana, so outside of Illinois. Thank you very much for including us in this conference. We appreciate the opportunity. Hope to hear from you soon. Thank you.
Thanks. We're going to hear from Corrine Daniels at Taylor Creek.
I'm Corrine Daniels. I'll be speaking about nursery production of climate resilient seed. I'm the nursery director. I have 25 years in the native nursery and ecological restoration business. Our nursery, Taylor Creek, is located about 60 miles east of Lake County and a stone's throw from the Illinois-Wisconsin state line. We're owned and operated by Applied Ecological Services, and we handle more than 600 species of local genotype seeds and plants. We're constantly adding to that list new to market and hard to grow species. And I'd say that's our passion. It's something that gets me personally excited about work each and every day. This is our primary growing facility. We grow more than 2 million native potted plants and thousands of pounds of native seed annually. We aren't the biggest, nor are we the cheapest native nursery in the region. That isn't our focus. Taylor Creek specializes in high diversity products with strict genotypic radius and quality requirements. We focus primarily on the Chicago region, but we also have growing locations in other regions. As a grower of natives, we've learned that our clients want diversity and they want local genotypes, but when it comes down to it, budget is king, and many will be forced to simply choose based on price. So we must produce at prices that can compete. Add to that challenge is ensuring that our products are climate resilient, and these are major drivers in the way we choose to grow. We believe that preserving wild-type genetic integrity leads to the best products and the most resilient restoration. And this requires us to grow and carefully track local genotypes, or if you prefer, ecotypes. And it means keeping our crops near to true wild type. If we're going to do that, we must maintain and improve the quality of our remnant wild areas. In the face of climate change, they are critical genetic arcs. We find it ideal to start seed production fields from remnant collected seeds, or as we call them, G0 or Generation 0, some would call them foundation seed. We expect that to result in a product that is more climate resilient. When we start with G0 seed, we know the true genotypic origin, and it helps us ensure that our products are as close to true wild type and all its genetic diversity as we reasonably can. We expect these true wild type are a hedge against many factors. Not the least of them is climate change. However, keeping it wild is a challenge. That's because inadvertent selection occurs at every step in the process. Accidental selection starts as soon as we collect foundation seed. It happens when we choose which remnant and where in that remnant to collect from. It happens when we choose which individuals to harvest and when we harvest them. At each point in our seed handling process, we risk losing the outliers. So we must manage the process to protect the outliers, not just the average. We expect the outliers will be critical in the face of climate change. Selection can occur in the greenhouse. It favors, of course, those which will readily germinate and thrive in the greenhouse environment. And wild types, as compared to cultivars, have less uniform germination, and germination can occur over many days, weeks, or months. Selection can occur when we install production fields, and some individuals will thrive under that cultivation, others will not. And the growing conditions at each nursery will favor some individuals over others, yet another selection. And it can happen when harvest occurs, particularly the harvest of large fields which are combined. Only those seeds which are ready that day will quite literally make the cut. For all those reasons, it's challenging to maintain an inventory with maximum wild-type genetic diversity, which we believe is the most climate-resilient product. It might sound like I'm making a case against nursery-produced seed. However, being aware of the danger of selection helps us to avoid it. When starting seed stands, we can collect G0 from plants over multiple areas of the remnant and over multiple days. We can harvest from the short, the tall, the early, the late, and so forth. We can be patient in the greenhouse, allowing for later germinating seeds to emerge. And after fields are established, we can reinfuse fields with G0 to keep the fields as near wild type as we reasonably can. We can harvest different areas of our seed production fields over a period of time, or hand collect in waves when seed is ripening less uniformly. We can avoid overhandling and over-refining our seed. Nurseries that employ these methods will never be the largest nor the cheapest. It's challenging and imperfect, and it does add cost. We understand our position in the chain of ecological restoration, and we have determined that these measures are worth it, particularly in the light of climate change.
That was really great. Thank you. It's time for our last breakout session. Thanks so much for sharing your thoughts. We have another set of lightning talks from Becky Collins from the Cook County Forest Preserves, Alyssa Nyberg from TNC in Indiana, and Josh Clark, who's with the Illinois Native Seed Network.
Good morning. My name is Becky Collins, and I am the Senior Resource Ecologist with the Forest Preserves of Cook County. If you're not familiar with the region, Cook County is located in northeastern Illinois, and our county seat is the city of Chicago. The map here shows our preserve system. In Cook County, nearly 50 different natural communities have been recognized. These diverse natural communities support approximately 1,200 species of native plants, just over half of the statewide total. Cook County is also the most populous county in the state and the second most populous county in the country. It's home to nearly 5.2 million people. Fortunately, 11% of Cook County remains in a natural state, most of which is protected by the Forest Preserves of Cook County, who own and manage over 69,000 acres of open space. The land that makes up the Forest Preserve system is a spectrum of land use history and ecological quality. We have remnant natural areas, some of which were acquired very early in our history. We have sites that are ranked grade A or B by the Illinois Natural History Survey and are high quality examples of 23 different natural communities. We also have former agricultural areas, woodlands that were heavily grazed by cattle or selectively cleared by settlers, as well as areas that were reforested by earlier forest preserve land managers. The forest preserves today also face threats such as fragmentation, off-site pollution, invasive species, excess shade, and the impacts of climate change. These threats necessitate ecological restoration and management, including the need for ecologically appropriate seed. All of this sets the context in which the Forest Preserve approved its first official seed policy in 2016. The policy was drafted by Drs. Brenda Milano Flores and Jeffrey Levin of the Prairie Research Institute, with input from Forest Preserve staff and a panel of local land managers, researchers, and volunteers. The policy outlines considerations and sets the preferred direction for obtaining and using plant materials. The policy also aims to balance our organization's need for seed with our desire to protect the genetic diversity found within our preserves. The policy includes considerations for using seeds such as matching soils, hydrology, and plant communities. It discusses genetic factors to consider such as inbreeding and outbreeding depression. For example, populations of fewer than 100 individuals may be at high risk of inbreeding depression, whereas those with more than 1,000 individuals may be at low risk. The policy also provides guidelines for seed movement, including best practices for seed collection, storage, and production in order to maintain and maximize genetic diversity. Ensuring diverse genetic resources provides plant populations with the greatest chance to adapt to climate change. Another key component of our seed policy is the use of seed tiers or seed zones. Our policy places a priority on local on-site seed and seed bank response when possible. If off-site seed is needed, Tier 1, which is Cook County, is the preferred seed source for use within nature preserves and remnant areas. Tier 2 is the Chicago Wilderness region and is the preferred seed source when Tier 1 is not available. Tier 3 is based on the concept of provisional seed zones and is the preferred seed source if Tier 1 or Tier 2 seed is unavailable. The seed zones included are located to the south and west of Cook County to account for anticipated climatic shifts in our region. Within Tier 1, Forest Preserve ecologists and volunteers have further refined our preferred seed zones based on sites with similar soils, hydrology, and plant communities. Shown here is an example of what this might look like in the Calumet area. We also abide by the Illinois Plant Translocation Policy for all of our state-dedicated nature preserves and land and water reserves. Currently, we are prioritizing areas for restoration that we believe have a potential seed bank or a strong volunteer seed program or specific project funds for acquiring and augmenting seed. We're also looking into creative strategies for meeting current and future seed needs while adhering to the intent of our seed policy. Thanks for your attention and have a nice day.
Good morning. My name is Elisa Nyberg, and I'm here in Northwest Indiana at the Nature Conservancy's Kankakee Sands property, where for the last 22 years we have planted 7,500 acres of ag ground to prairie. Since 2016, we've had bison grazing 1,100 of those acres. My roles here over the past 20 years have included managing our on-site nursery, sourcing seed and plant materials, and overseeing our outreach efforts. When Kankakee Sands was first envisioned back in 1996, we dreamed that it would be a seamless connection among the existing natural areas present in Newton County, Indiana, and Iroquois County, Illinois, that encompass more than 25,000 acres of sand prairie, sedge meadow, marsh, pin oak flatwood, black oak savanna, and dry sand dune communities, and host at least 86 rare, threatened, and endangered plant and animal species, if not more. And for that reason of wanting to be a seamless connection, it was critical that our restoration acres be of the right species, the right diversity, and the right genetics. Our species and seed protocols have changed very little over the past 20 years. Initially, our seed range was a dot on the map with Kankakee Sands at the center and a 50-mile radius. We revised our seed protocol, avoiding geopolitical lines and using the filters of distance, community type, and soil type. But even with this strict protocol, we had a gray area. Occasionally, a species would come up that we would debate long and hard about whether to include or not include. Hard discussions, but important ones. Our Kankakee Sands timeline was to restore 500 acres per year for 10 years, and we would need about 2,000 pounds of local genotype seed per year. The species diversity in the ecotype that we needed was not available for commercial nurseries. So we came up with a two-pronged in-house approach, create a Kankakee Sands on-site native seed nursery, and develop a wild seed collection program. The nursery was established in 1999 to grow 150 species conducive to growing in a nursery environment. It had an expected life of 10 years, after which it would be decommissioned. Each bed at the nursery was created by using the seed of plants from 5 to 7 different locations to ensure genetic diversity. We were able to grow plant stock from our nursery beds in our own greenhouse. We had the ability to harvest mechanically, irrigate, fertilize, manage pests and weeds, and conduct prescribed fire, all for maximum harvest. We were able to procure more than 1,500 pounds of seed per year with a budget of $80,000 to $100,000, most of which was for staff salaries. To round out the many species that did not grow at the nursery, as well as maintain a diversity of genetics, we developed a wild seed collection program. Over time, 250 seed collection sites were established on both private and public land. The wild collection program contributed significantly to our plantings despite the challenges of long travel times, the uncertainty of the harvest, and the pressure put on the wild populations. Stephanie Frischi, pictured here with Greg Livovich, managed the wild collection program at Kankakee Sands for many years and was instrumental in helping with this presentation. Our harvest protocol has been to harvest as much as we need from our on-site seed nursery, up to 100% of the seed of a bed. But for the wild collections, we harvest no more than 1/3 of a perennial species and 1/10 of an annual species. A quick mention that we do process our own seed on site and mix our seed batches as well. The seed mixes for our early restorations until 2016 were relatively high diversity, with some years plantings including as many as 400 native plant species. In general, our nursery production made up about 2/3 the volume and 1/3 of the species diversity of the seed mixes, and the wild collection made up about 1/3 of the volume and 2/3 of the species diversity. 2016 was a pivotal year. The nursery had been useful for more than the projected 10-year timeframe. We thought we were nearly finished with our restoration plantings, and it was now time to divert time and funding to managing the 5,000 acres that we had in prairie. Our nursery and wild seed program were effectively shut down. However, since 2016, we have continued to do restoration plantings, which range in size from 60 acres to 320 acres. Seed diversity and local genetics are still critical to our work, and we are now left to scavenge what we can from the nursery, collect with permission and respect on neighboring properties, and purchase local genotype seed from nurseries. We're so grateful to nurseries that do carry local genotype seed. As we continue our plantings without access to a functioning nursery, we struggle with the thoughts that after so many years of heavy harvesting, the nursery has certainly lost much of the original genetic diversity, and are concerned about the pressure that we put on our own restoration plantings and the wild populations when we harvest from them. In retrospect, I wonder if we underestimated the value of our native seed nursery, not only to our own work, but to providing local genotype seed to our partners in the Kankakee Sands area. I think there is great value and will be an increased need for commercial nurseries to provide local genotype seed in the near future, perhaps a farm-to-table-like program where the grower and the purchaser know one another well and work together to strengthen conservation work in their local area. Altogether, our seed strategies include the following. Diversify the seed mix and include as many species as historically present, including those annuals and early successional species which assist with recolonization when disturbance occurs. Provide for a diversity of genetics within plant species by harvesting from multiple populations. Modify water levels at the planting site to create wet areas that are deep enough to withstand extreme drawdown, and create a way for mesic areas to stay mesic and the wet areas to stay wet. Include planting mixes that span hydric zones to allow for hydric fluctuation over time, like xeric mesic mixes, mesic wet mixes, and wet emergent mixes. Overseed areas that need genetic or species diversity, do not establish well, need bolstering from invasives, or experience disturbance from drought, flood, or grazers. Document the plantings via a database listing each planting unit and each seed lot that was planted, allowing us to track not only species but also genetics in each planting. And hope like crazy that it works. If you'd like to come for a visit or talk more about our seed program, please reach out to me. I'd love to chat more. Thanks so much.
I'm the restoration ecologist with the DeKalb County Forest Preserve, and I'll be talking about the Northern Illinois Native Seed Network today. Ed Cope was our founder. He had worked at NLI for several years. He had worked for Winnebago before that. Moved over to NLI and was surprised to see the difference in seed resources. Asked around, found out, yeah, this will be a good idea, so he started the group. Ed's initial ideas were to share locally rare and uncommon seed, work together to restore historically common species that have declined, share seed resources that may be common for some but not others, and share seed collection, processing, storage, mixing, and knowledge and ideas about seed. Some of the agencies that are part of our group, there's others, but this is the most of them. So it's nearly 20 agencies involved to one degree or another: Forest Preserve, Conservation and Park Districts, nonprofits, charitable organizations, land trusts. We've also picked up a few private landowners, organizations varying size, structure, funding, and land holdings, but all have similar goals and visions, and they all want to share their essential resources, seed and knowledge. Potluck in the late fall, early winter, and we share rare species. So a fraction of an ounce, give it to somebody else, they establish population. When their population is producing seed, they can bring that in and share it with the rest of the group. Hard to collect species, stuff everyone might have, but difficult to collect. So by combining resources, one agency might be able to get a population started and then eventually kick back into the group. And then bulk seed, things you get a lot of, yeah, bring it in, you might be able to trade it for something more interesting from someone else. Our past local species have been Chilone glabra, turtlehead, and GM triflorum, prairie smoke. This year, 1920, it was the Hypoxis hirsuta, yellow stargrass. Once a lot more common in the prairies. Steve Packard has a nice blog about it on his blog. Agencies contributed seed given to 4 members to propagate. One agency, Lake County, grew enough plants to share. Thanks, Luke and Kelly. All plants were distributed were of mixed sources. So didn't all come from one agency, diversity there. And then plants were distributed to 9 different agencies to establish populations. And I established a seed source bed at our natural resource shop. More agencies develop populations that can be picked and shared that we kicked back into the group. So you get some going, you can bring it back in and share it. That I established at Afton Forest Preserve. And we've gotten good seed out of it every year. And some plants that have ventured out into other beds have been moved to places out in the prairie. So how are we responding to climate change? Increasing access to seed from plants adapted to our region. So more access to plants that have adapted to northern Illinois climate. We're also increasing knowledge on seed collection, storage, processing methods across the region, so that as people collect more seed or are wondering what to do with their seed, their efforts are successful, and then that helps the group as well. And then we're increasing genetic diversity across the region. So taking some plants from one site that may be genetically a little different, bringing it to another site, putting it in a planting there, mixing genetics a little bit from the region, hopefully increasing that diversity and not getting into like genetic bottlenecking. To address climate change, not something we've discussed a whole lot, but something we feel, like the rest, like everyone else, that we need to address. So would it benefit us to get seeds from hotter or drier climates? Things from a little further south of our region. How would they fit in genetically? And then expanding access to plants that have a wider range of tolerance and are more resilient. So tolerant to drought, tolerant to heat, more resistant and resilient. If they live on the edges of wetlands and are adapted to dry downs, maybe they would do better in a drought situation, kind of like we had this year. And then focusing on rare or uncommon plants that kind of have those tolerance and resilience and resistant traits, and figuring out which ones those are and growing them in the group and making them available. So I will take questions after this. Thank you very much.
Thanks so much to our last group of lightning speakers for providing their talks. I really want to thank all of you for attending today. I also really, truly want to thank our speakers for taking the time to put together some really terrific presentations. It was a lot of work and they were superb and highly informative. Really appreciate your thoughts and your time. On behalf of the organizing committee, I really want to thank also the facilitators who led the breakout groups for their time and efforts. Thank you to everyone who attended for your time, your attention, and your thoughts. We hope that you found today interesting, informative, and enjoyable, and hope that it was a productive use of your time. Just want you to know that if you're interested in joining a working group, we want to keep this conversation going. Clearly, we've only just scratched the surface today. It's an issue that is really important. I think we all are asking the same questions. I think we all are tending to find some similar themes and perhaps some similar solutions. And it would be great if we can collectively try and take advantage of all of that and maybe come up with some kind of a regional approach to thinking about how to solve this problem. So please feel free to email any of the co-organizers or myself, and we'll continue to set those meetings up. The next one that is on deck will be at Wild Things in February. Thanks again, and I hope you all have a safe and wonderful rest of your day.
And we should say big thank you to Patty, who has rounded everyone up and has been the directing and managing and pushing everyone forward, and has thought of this and went out and got a grant for this. So thank you, Patty.
Really my pleasure. This is a issue that is near and dear to my heart, as most of you know. So thanks again. I appreciate that. Bye, everybody.